Thank you all for coming. We're all falling asleep. Wake up! No, I'm just kidding. My name is Tommy. I'm a PhD student at UC Berkeley. Today I'm going to be talking at you guys about patterns of host pathogen diversification among deer mice and Sumeryra hantavirus. But before I get into it, I'm just going to start by going over the broader significance of this work. And so here listed are just some of the most common human infectious diseases that have caused epidemic and pandemic events in recent time. And one thing that all of these have in common, I'm sure you recognize them, is that they're all zoonotic diseases. So they came and spilled over to humans from other species. And currently, 3 out of every 4 newly emerging infectious diseases are zoonotic in origin. And this number is increasing through time, along with the number of spillover and subsequent outbreak events through time, especially through the 20th and 21st century. And so this is largely due to factors that all relate back to anthropogenic environmental changes, whether that be to the climate or to the landscape, right? And so really now more than ever, it's important that we not only understand the interactions that are happening between zoonotic diseases and their reservoir hosts, but we understand the levels of association that are going on between them, as doing so can allow us to ultimately be able to predict and manage for outbreaks when they happen, and just understand underlying mechanisms driving spillover and transmission across host pathogen systems. And so on that note of understanding host pathogen association, understanding the level at which a pathogen is associated with its reservoir host is important because strong interactions between the two can lead to co-evolutionary mechanisms that ultimately drive pathogen diversity as they diversify alongside their hosts. And that increased diversity results in novel variants with increased likelihood for host switching events, that's between wildlife species, but also spillover events from wildlife to humans. And so this point is particularly relevant when it comes to hantaviruses. The reason being is that while hantaviruses are super diverse, one thing they always have in common is that they're associated with a single host species, so setting the stage for these types of co-evolutionary drivers of pathogen diversity. And another thing with hantaviruses is that they're segmented RNA viruses, which means that they're able to undergo gene flow through processes of recombination and reassortment as an additional source of diversity that can otherwise drive host switching and spillover events. Now, hantaviruses are globally distributed, but by far the most deadly hantavirus that exists with respect to virulence is Sumeryra virus, which is endemic to the western U.S. As you can see, all those red states of human spillover across the western U.S. And so, while in the reservoir host, the rodent that hosts Sumeryra virus, it doesn't cause any disease. When it spills over to humans, it has a case fatality rate of 50%. All right, and so to put that in perspective, that's like well over 10 times the case fatality rate of COVID-19. So spooky stuff. But it's when it spills over to humans, it does so through airborne inhalation of airborne feces and urine from rodents. And unfortunately, there is no known cure, vaccine, or treatment for anyone infected with hantavirus. So the best protection is to avoid areas and situations that have a high probability for spillover. And so the way that we have classically gone about understanding spillover in this system is by understanding the reservoir host, the deer mouse, which I'll be referring to as Peromyscus maniculatus with subspecies labels. And so particularly understanding the contact zones between subspecies of the deer mouse, which are shown by those different colors on the left map. And so the reason being is because it's thought that the contact zones will represent areas of gene flow among the virus, and as well as obviously between the host. But this model assumes, of course, that the host and the pathogen are co-diversified, or in other words, that distinct subspecies of the deer mouse will carry distinct variants of hantavirus. And so if we kind of look on a broad scale, these major contact zones between subspecies in the western U.S., we see that they align pretty well with the states where we see the highest human spillover. But nonetheless, this baseline hypothesis of co-diversification has never been tested in this system, just largely due to a lack of genetic data for both the host and the pathogen. And so that is what I'm aiming to do with this project, is simply show, do we see genetic co-diversification aimed across the state of California, as we have multiple lineages of deer mice, along with a high propensity for spillover into humans across this state. And so to do this, essentially what I'm going to be doing is saying, can we first just test the subspecies designations of deer mice with whole genome SNP data across the state? and then delineate the genetic structure of Sanal virus, as this virus has gone historically really just undersampled from a genetic perspective, and then look at phylogeographic signals for co-diversification between the two. And so with the baseline prediction that not only should we see co-diversification taking place, but those contact zones between host and pathogen populations should provide some context and some insight into host switching events as well as spillover across the state. And so now I'm just going to go briefly over the background that we understand already for the diversity of deer mice across the state. And so people have been studying deer mice for a long time, since the early 1900s. And so there was originally 3 subspecies identified across the state just based on morphology alone. And this includes Peromyscus maniculatus rubidus, Sonoriensis, and Gambellii. And then we also have subspecies that are thought to be associated with each of the Channel Islands off the southern coast of California. All right, then more recently, people have come with mitochondrial DNA and restructured and condensed these subspecies across the mainland to basically just exclude this rubidus subspecies at the top, leaving us with Sonoriensis and Gambellii subspecies with that north-south contact zone across the state of California. And so the data that I'm using to assess the subspecies of deer mice comes from the California Conservation Genomics Project. And so this includes 91 samples, all taken from specimens from the Museum of Vertebrate Zoology. And we've done whole genome sequencing with SNP data of over 400 million SNPs, but for the results I'm showing today, I've thinned that down just to 1 million SNPs to make it manageable for a computer. And so again, we have samples from the mainland as well as from the Channel Islands. And so if we just take all those samples and just start by plugging in into a principal components analysis, what we get is 4 genetic clusters popping out. And not surprisingly, the most distinct cluster belongs to that of the Channel Islands, as they've been isolated and separated from the mainland for quite some time. And then we have the mainland samples down here. So, just for time's sake, I'm going to kind of exclude the Channel Islands. We understand they're very distinct, and then dive into these mainland samples right here. So if we zoom into those mainland samples, what we get is this picture coming out. 3 genetic clusters that are all corroborated by admixture as well. And if we plot all of these data points across the map, we see they're all geographically distinct across the state of California. All right, and then we can kind of turn back the page again to what we already know about deer mice diversity across the state to make sense of this phylogeographic pattern. And when we do, we see clear support for the mitochondrial lineages of Sonoriensis and Gambellii subspecies with that north-south contact zone. And then if we turn back the page a little bit further for this other blue subspecies in the northwest, we see that that aligns perfectly with what was originally identified as the Peromyscus maniculatus rubidus subspecies in the northwest corner of California. And so with that, we're left with this really nice picture where in the north, between rubidus and Sonoriensis, we have them separated by the Cascade Mountains. And further south, we see that levels of admixture actually positively correlate with increasing elevation across the Sierra Nevada mountain range, suggesting that this mountain range represents a barrier between these 2 subspecies and a point of contact between them. And so that's our kind of story for the deer mice. And so now we're going to go over some background for Sanal virus across the state. Starting with our understanding of human transmission, human spillover, and see what we see in trends of human infection. So across the state, the highest percentage of human spillover occurs across Sierra Nevada. And this is interesting because we don't see any sign that spillover actually correlates to human density, population density, as you might expect. In fact, those 3 counties across Sierra Nevada that have the highest spillover incidence are actually only account for 0.1% of all the California population. There's almost like nobody living over there. All right, and we also see that actually cases positively correlate with increasing elevation across the Sierra Nevada mountains. And furthermore, there's been human spillover across most of the state, but except for this region in the northwest corner, where we haven't really seen any incidents of spillover, and we've hardly even detected Sanal virus across that region to begin with. And then if we're looking at deer mouse seroprevalence, we see similar patterns. So we see across Sierra Nevada mountain ranges where we see the highest seroprevalence across deer mice populations, with the second coming in the southern coast. As far as genetic diversity goes, so this is just currently the only published data of genetic data across California. And so that whole orange bar just represents the Sanal virus across all of the US. And then right here in that little box is the clade of California samples And we can't really say much based off this data other than there seems to be signals of genetic structure across the state. But one thing that we can say that's very interesting is a scenario of a host switching event, the only host switching event that's been documented for Sumeryra virus, where the virus jumped from Paramyscus maniculatus to a closely related species, Paramyscus eremicus. And this is localized in this region of the Death Valley of California. It hasn't happened anywhere else. And I'm going to come back to the significance of this host switching event in a second. And so now going into the actual data I used to approach this virus, I've collaborated with the California Department of Public Health, and they provided 94 samples with less sampling across geographic space, but we have gotten sequencing of the medium genomic segment, which is the glycoprotein precursor. It just means that that's the region of the genome that's interacting with host cells. All right, so that shows host specificity. And this is sequenced from deer mice tissues, and they came from different individuals from the actual deer mouse data I showed you before, so that's worth pointing out. And so again, mainland and Channel Island samples are both included in this data set. And so if we just plug that in and look at the phylogeographic distribution, so we see 3 regionally distinct clades. The first one at the bottom associated with the Channel Islands, and then we see a clade associated with Southern California in green, and then kind of northern, northeastern California in the orange. And this northern clade in the orange can be further broken down into multiple geographically overlapping clades that are all distinct, that are all concentrated in that region of the Sierra Nevada area. And also embedded in this data set are those samples taken from Paramyscus eremicus, the other species where it switched hosts down in the Death Valley region. And that again groups in with that northern clade and that orange clade in northern, northeastern California. And so now kind of coming back around and bringing the whole story together, and we walk through it, we see that we have lineages for both the deer mouse and Sumeryra virus associated with the Channel Islands. We also see lineages associated with Southern California, represented in green, and the northern, northeastern California region of Sierra Nevada. And the only area where we don't see a direct correlation between this host and pathogen lineages is in the northwestern part of the state, where we see the Rubitus subspecies. But that's just because we haven't actually been able to detect samples of the virus across this area. So that could be maybe the virus, the subspecies lost the virus, or it's just occurring such low prevalence that we haven't been able to detect it. But again, this is also an area where we haven't seen any spillover happening. So from this perspective, it seems like it's a relatively safe region to be in. And then so we see that the major contact zone then occurs right across the Sierra Nevada with this region where we see that major contact zone between the host as well as the virus. And again, this is the same spot where we observe increased genetic diversity for Sumeryra virus. And it's also across that same contact zone that we see that host switching event also taking place, suggesting that possibly gene flow might be driving diversity across this region between those 2 variants, north and south of Sierra Nevada, which could have facilitated this host switching event as well. And then now again, we also see in the same region of increased diversity in host switching a higher incidence for spillover for humans across the state. And with that also just higher seroprevalence of deer mice in general, strongly suggesting that this is a real hot spot for interactions. And so now coming back full circle, asking the question, do we see co-diversification taking place? It does see that we see strong signals for co-diversification across the state of California, with deer mouse lineages corresponding to Sumeryra virus lineages. But more so than that, what we see is that major contact zone between host and virus lineages is located right across the Sierra Nevada mountain range. And this is also where we see increased Sumeryra virus diversity. We see increased seroprevalence across deer mice populations, high incidence for human spillover, and again, a documented host switching event taking place right across the contact zone, all suggesting that this region is a really, really important area for emerging eco-evolutionary dynamics between deer mice and Sumeryra virus. And so future directions for this project, I'm currently using ecological niche models to explore environmental drivers between host and pathogen lineages and see how they might respond with future environmental change, as well as assessing genomic patterns of coevolution by looking at signals of selection across the genome for deer mice, or possible regions that would explain why the mouse just doesn't feel sick when it gets the virus. It's completely tolerant to this pathogen. And then also increasing genomic sequencing for Sumeryra virus to assess historic reassortment and recombination events across the state. And of course, expanding this analysis outside of California so we can have a holistic perspective of emerging dynamics across the whole Western US for this pathogen and for this host. And so with that, I would like to thank all of my funders, as well as everyone at the California Department of Public Health and everyone in the California Conservation Genomics Project. Shout out to my lab who's walking out. I'd tell you to go see their presentations, but they already gave them. All right, and so if there's time for questions, I'll take them, but if not, it looks like we're time to switch over. All right.
Let's start. Okay. Good afternoon, everyone. Welcome to my presentation. I'm talking about Tabanus bromeus. Tabanid family, cosmopolitan medical and economic importance. Horsefly of Tabanid family are common blood-sucking pest of mammals. Bromeus is one of the most common pest species and medical importance distribute Palearctic region, including Turkey. And despite its medical and economic significance, apart faunalistic studies, there have been no details study Bromeus Turkey, other countries. Turkey West Palearctic region is highly significant for diversity and numerous endemic species. Anatolia is known for high hybridization due to topography. It is homeland of many species and refuge area those species affected geological and climatic change in the past. It has much more biological importance than many other places in the world, considering its role as natural bridge Europe, Africa, and Asia. Bromeus is not only subject significant curiously, but also vector of various diseases, making its study crucial for public health and economic reasons. We want to determine population genetics and demographic history and phylogeographic patterns for economic importance. By analyzing the genetic diversity and distribution of these species, we aim to uncover how past climatic and geological changes have shaped its current population structure. For this purpose, we collected, using Malaysia tramp, we collected 13 populations in Turkey and 2 populations, Croatia and Iran. DNA isolation was done. I used fresh material and museum material. Then amplified cytochrome oxidase 1 and ITS region by PCR. After that, analyzes data. ITS consists of ITS1 and 5.8S RNA, ITS2, 5.8S RNA show no polymorphism. We eliminated it from our data. ITS1 and ITS2 we concatenate and data analyzed. You can see genetic diversity. Sequences cytochrome oxidase gene 233 individual generated 169 haplotypes. ITS 247 samples collapsed into 990 alleles. When we consider two biomarker together, Tabanus bromeus show quite remarkable genetic diversity populations. Demographic history. The mismatch distribution analysis and other population demographic analysis, FUIS, FS, TAJIMSD, HIRI, SSD, suggest rapid demographic expansion from a small population in the past. The high haplotype and low nucleotide diversity bromeus, cytochromoxidase and ITS region also supported demographic history. I did FST and AMOVA analysis for population differentiation. Geographic structure across the distribution range of the Tabanus bromeus in Turkey is further supported by the pairwise FST and AMOVA analysis predicted the levels of genetic differentiation of populations and reveals significant difference from each other many populations. Phylogenetic and phylogeographic analysis. All the three analyses conduct for the both data set produce similar tree topologies with different boost probability value. Thus, only Bayesian consensus through cytochromoxidase and ITS data. Bromeus haplotypes split from outgroup around the Miocene period, you can see. Results indicate from the structure bromeus genetic lines was significantly influenced by climatic and environmental late Pliocene and early Pleistocene period. Phylogenetic analysis shows species appears to enter Turkey, Caucasus, and Iran, and then the geographic barriers such as Anatolian diagonal. Network analysis supports general pattern phylogenetic tree cytochromoxidase gene. ITS data, phylogenetic tree topologies and network analysis also support this cytochromoxidase result. Due to presence of the Tabanus bromeus species in countries such as Russia and Ukraine, it may be assumed that the lineage north of the Black Sea, which was freshwater lake at that time, reached Europe. Moreover, these results agree with our findings that the Turkish Tabanus bromeus population originated in the Turkish population is Artvin and Iran. Let me see. For more detailed information, as you say, thank you for your listening. I am excited.
Thank you for being here. I'm Xavier. I'm a PhD student in Université Laval, that's in Quebec City, not too far away, and I'll be presenting our phylogeographic study in the high Arctic using whole genome sequencing. So obviously, around the globe, everywhere in the world, biodiversity has been impacted heavily by climatic oscillation in the last glacial era of the Pleistocene. And perhaps nowhere as drastically as in North America, where basically large majority of present-day Canada was covered by ice sheets 20,000 years ago around. And obviously that created some drastic changes in distribution ranges of pretty much every species. That creates bottlenecks and contribute to the diversification of lineages that might have survived in different glacial refugia. And obviously when the ice melts, those lineages will likely recolonize the newly available habitats, which contributes to second contact between those diversified lineages. And the field of phylogeography has been studying this for a while now, starting in the 90s with mitochondrial haplotypes that have an evolution rate that allows for identification of those lineages, mitochondrial lineages at least. But then with advances in technologies, we have been introducing the nuclear genome also in phylogeography, which kind of blurred the line with the population genomics. And in recent years, there's more and more whole genome data sets available. So we can ask ourselves, what do we have to learn and to gain from this newly kind of resolution? And obviously, we can think about the mitochondria and the nuclear genome as complementary in our understanding of biodiversity, because obviously they are inherited in very drastic different ways and evolve separately. So in mitochondria, as you know, is maternally inherited, whereas in nuclear genome, when you have two, for example, those blue and yellow lineages that meet, you'll have, through generations, with recombination, a mixing of those two lineages and subsequent admixture that breaks down toward an admixed genome. But obviously, that's considering that the recombination is homogeneous in the genome, and if we have some parts of the genome that don't recombine or recombine less than elsewhere, for example, in chromosomal inversions, we might have some parts of the genome that are actually protected from recombination, and that could keep some haplotypes that we can trace back from the ancestral line. So obviously that creates a mosaic of different local ancestry tracks of different length that can arise from all those stages of admixture. So here I studied Arctic char, which is the northernmost freshwater fish. I think it's very interesting in its distribution. It's all over the world, all around the globe, with obviously has been drastically impacted by glaciations. There's a lot of different lineages that do meet up in, all around its distribution. It is a phylopatric and sometimes anadromous fish, as you can see in the red distribution, meaning that it goes to sea to feed and comes back to fresh water. So there's a lot of dispersion potential also. And fishes are known to have frequent intra and interspecific hybridization. And just before moving forward, I just want to shift a bit our expectations about glacial refugia when we are so high in the Arctic. So obviously we oftentimes think about glacial refugia at the south of the ice extent, but the ice didn't really reach all the Arctic, and there has been some evidence in different taxa that there might have been some cryptic refugia in the high Arctic. So here we gathered a huge collection of Arctic char from 33 populations, distinct populations here, that we sequence with low coverage whole genome sequencing. So there's a lot of interesting questions that we can ask more locally in this data set, but I'll focus on more broad questions today. So we achieved coverage of 2x for this data. And we ask ourselves what can we learn about the evolutive history of Arctic char in Arctic Canada with this newly acquired whole genome data. But good thing about the whole genome data is that you also get mitochondrial DNA for free. So we started there and identified those two lineages, the Arctic and Atlantic in yellow that were already described in previous studies. And so they diverged a while ago, and their present distribution is as we can see here. So we think we covered most of the distribution of the Arctic lineage in blue, and we see that it co-occurs with the Atlantic in the eastern part of our populations. Note that we also have an outlier population, an outgroup population in here that is actually a lake in Sweden. So that is also from the Atlantic lineage that we think actually comes from Europe. And then we jumped in the nuclear data. We use, oops, is it working? Yes, spoiler. Oh, it's a bit laggy, but. So we use ANXD to genotype a bunch of SNP that we filter heavily. I'm skipping a lot of steps here, but salmon is a very complex genome, so there's been a lot of filtering in paralogs. If you have questions about that, I'm happy to talk about it later. But we started with looking at admixture patterns in the nuclear genome. And what we found with our Swedish samples also is actually a lot more admixture that we saw in the nuclear genome. So we have this whole southern Arctic, Canadian Arctic, that shows a signature of admixture in the nuclear genome. If we look at this in a geographical standpoint, starting from the westernmost population and measuring marine distance, we see this very clinal pattern in average whole genome ancestry, with those southern populations being like the putatively admixed ones here. We also saw that heterozygosity and nucleotide diversity was higher in those intermediate admixed populations. And so we think that's was following also F4 tests and ABABABA and a bit of demographic modeling. We think that's several lines of evidence for admixture at the nuclear level between the Arctic and Atlantic lineage. So that leaves us with major discordance. I feel like my slides are loading slowly, but it's coming. Okay. So that leaves us with a major discordance between our mito and nuclear genomes, where we have this whole part that is that we think is admixed between the nuclear genomes of those two lineages, but we don't see any of the Atlantic mitochondria in those populations. There might be a lot of few interpretation of this. Obviously, it's maternally inherited, and there has been sign that male Arctic sharks do migrate or do disperse more than females. Maybe that has something to do with it. Obviously, mitochondria is haploid, so has a lower NE, but what I think is most interesting is that maybe there could be selection against the Atlantic mitochondria in the Arctic environment. But we wanted to dig a little deeper with our whole genome sequencing data, and we wanted to look at what it looks like at the nuclear level, this hybridization at the finer genomic scale. Basically, can we use our data to find those local ancestry tracks, haplotypes, haploblocks that tracks from those ancestral lines? And we borrowed a method that is used more and more to identify large structural variations, that is local PCAs. So you basically scanned your whole genome doing PCAs all along small windows and are looking for consecutive windows that show a very distinct pattern of genetic variation as the rest, and that you could interpret as non-recombining haploblocks that will create these three very distinct groups with the two homozygotes, for example, for an inversion, and the heterozygote in the middle. So we actually did that and found a bunch of them that are maybe less clear than some that you've seen before, but I think are pretty convincingly that there is something happening on this PC1 that explains a lot of the genetic variation, and that we kind of attribute to those AA, AB, and BB genotypes. What is interestingly is that it really strongly fits with our second contact scenario, with those two regions at the far west and east being purely homozygotes for those blocks, and those haplotypes meeting in the middle. We find a bunch of other mores that are all independent, but all share the same geographic distribution in allele frequency. We think that by scanning our genome, we found up to 22 independent putative local ancestry tracks that are fairly big in this case. And one last question that we wanted to ask is, so we've been interested a lot in this Arctic lineage that is covering the whole Canada, but can we actually pinpoint where it might be coming from? Where is the putative local refugium? So we use time difference of arrival at locations that uses ancestral and derived alleles proportion that we polarize using data from sister species to know which allele is potentially ancestral and derived. We use the directionality index between all pairs of populations, and it becomes a kind of triangulation of where, assuming that derived mutations accumulate along the recolonization route, can we triangulate the potential origin of this recolonization. And here, what it looks like when we take the whole genome, we have, at least in here, strong evidence for an origin of recolonization from the south of our study range. But considering that we now know that this is a two-origin system with the Arctic and Atlantic lineage meeting in this part that is maybe inferred as a potential origin, can we really trust this inference? So we're thinking about how can we maybe dissect the signal and focus only on the Arctic lineage, that was our primary focus. So we tried basically removing introgression from the Atlantic from the dataset. So we went back to our putative local ancestry tract, those blue individuals, for those specific part of the genome that we know should carry mostly genetic variation that comes from the Arctic lineage. And we repeated the same analysis using only them. And we made a bunch of replicates, and the story is very different. It's very less clear. There's a lot of variation between runs. But what is interesting is that if we take random regions that are also small as our local ancestry tract, we actually find the whole genome distinct pattern. So we think that focusing on genetic variation inside those local ancestry tract could help us contrast the story of our two lineage. So just as a summary, so we identified this massive post-glacial hybridization and strong mitonuclear discordance in our data. We also explored how we can use whole genome data to identify at least bigger local ancestry tract, even if it's at low coverage. And I think that makes us think on how maybe the most interesting part of having whole genome data is not necessarily that you have like millions and millions of SNP. It's maybe more the horizontal signal that you can get out of it. How are those SNPs linked, and how that can be used to basically investigate more complex post-glacial histories. And with that, I want to thank my amazing team at Laval and everyone that made possible the analysis and sampling and sequencing. And thank you. I think the gong hasn't gone, so if there's questions, yes?
Really interesting talk. I'm really interested in your mitonuclear discordance work.
Yes.
Selected against hybrids.
Yeah.
Would you mind elaborating a little bit on like possible fitness consequences abilities in this system?
My feel from this data is that there's not a lot of selection against hybrid per se. It just seems that it's the same story is not happening for the mitochondria and the nuclear. So I think maybe it's mostly the mitochondria, the Atlantic, the yellow one that is selected against. We know there's a lot of important functions on the mitochondria, and the environment are very contrasted between the Atlantic. But as like hybrid depression, it doesn't seem to be the case. Those two lineages seem to be inbreeding for a while now, and doesn't seem to cause any problem that we can see from this data at all. But thanks.